Hello everyone. Today in this physiology video lecture class, we shall be discussing regarding the hormonal control of metamorphosis in insects and amphibians. Now regarding metamorphosis, we all know very well a larva undergoes series of structural and physiological changes resulting in formation of an adult. Okay, there are so many changes that are taking place. There is formation of the new structures. At the same time, there is disintegration or disappearance of the previously existing larval structures. Now this process has to be done in a regulated manner. Once the adult structures are formed, the larval structures has to get disintegrated, and this phenomenon has to happen in a controlled manner. Now, who is controlling all these activities? The hormones are controlling. Okay, so in this video lecture, we shall try to understand, especially in case of insects and amphibians, how exactly the hormones are playing their role in order to bring about a successful metamorphic changes to form a sexually matured adult. Okay, so let us get started. Now, you see, metamorphosis means, as I have told you, it is a complete transformation from an immature larval form to a sexually matured adult. Okay. Now, this may be a change of morphology, this may be a change of function or habitat. All the three changes are involved here. As we are concentrating on regulation or hormonal influence on the metamorphosis, we know very well that the hormones are secreted by the endocrine glands. Now we shall see the hormonal regulation of metamorphosis in case of insects and amphibians. Now in case of insects, we know very well the insect undergoes series of molting. Molting means the egg dices are casting off the old skin, and this casting off is after the formation of the new skin just below the old skin, and this results in formation of imago. Imago is the final stage of metamorphosis. Now, in between insect larva to an imago, there are a lot of structural and physiological changes that are taking place, and all these changes are completely controlled by different glands. Now, in case of insects, which are the glands that are involved in regulating the process of metamorphosis? Now we can see here brain, prothoracic glands are there, and corpora allata. These are the three important structures that are involved in regulating the metamorphic changes in insects. So in this flowchart, you can see now the brain secretes during the metamorphic process. The brain secretes an hormone known as the prothoracotropic hormone. This prothoracicotropic hormone, it influences the prothoracic gland. Now, the prothoracic gland secretes an hormone called ecdysteroid or ecdysone. It is also called ecdysone, and brain also influences another structure called corpus allatum, which is present attached to the brain, and this secretes an hormone known as juvenile hormone. Both these hormones play a very, very important role in bringing about metamorphic changes. So let us see one by one. Now, the same flow chart has been depicted in a different form here. Here there are neurosecretory cells that are present in the brain. They produce the brain hormone. The PTTH or prothoracicotropic hormone is called as also called as brain hormone. This acts on the prothoracic gland, and prothoracic gland produces ecdysone or ecdysteroid, and this acts on different cells of the larva and promoting their growth to result into an adult. Now, at the same time, the juvenile hormone that is produced by corpora allata, it tends to maintain the larval characters. It is important, remember, students, unless there is a good situation of formation of the adult structures, only then there should be disintegration of the larval structures. The larval structure should not get disappeared unless there is formation of perfect adult structures. If there is imperfect formation, if the larval structures also disappear, then the larva tends to die. So, for that reason, the juvenile hormone tends to maintain the larval characters and avoids earlier development or improper development of larva into adult. In fact, to say the juvenile hormone maintains the quality of metamorphosis. Okay, it tests the quality of metamorphosis, and once there are proper structural changes that are taking place, the levels of juvenile hormone keeps on reducing, and there is formation of the adult. So in this flowchart, you can see there is equal amount of the ecdysone that is produced at all stages, but you can see the juvenile hormone, it is highest in the larval cells, and it keeps on reducing from the larval cell to the adult cells. Now, as I have told you, the brain secretes the brain hormone or prothoracotropic hormone. This influences the prothoracic gland prothoracic gland, and the prothoracic gland produces an hormone called molting hormone or prothoracic hormone. This molting hormone is very, very important to bring about the changes from larval form to the adult. Apart from that, the neurosecretory cells of the brain, they also pass through this corpora allata, and the while passing through it, the hormone that is produced, the brain hormone that is produced by the brain cells, neurosecretory cells, will be passing on to these structures. Now, what about the prothoracic glands? The prothoracic glands are stimulated by the PTTH that is produced by the neurosecretory cells of the brain, and stimulation of prothoracic glands results in formation of the ecdysone. This is the molting hormone which initiates the growth and the process of molting, resulting in shedding up of the old skin, and there is successful formation of the new skin. Apart from the brain and prothoracic gland, corpora allata plays an important role in controlling the metamorphosis. It produces a very, very important hormone known as juvenile hormone. This juvenile hormone, as I have told you while discussing that flowchart, it controls the qualitative change. It makes sure there is proper formation of the adult structures, then only the larval structures start disintegrating. Once there are qualitative changes that are taking place, the levels of JH reduce. Okay. Otherwise, if there are no proper changes that are taking place, the levels of JH will not reduce. As a result, the larva may become a giant larva. It will not get converted into adult. If the larva has to become adult, all the adult structures has to be properly formed. It is made sure by the JH. Okay, and these structural changes that has to be properly occurring, it will be controlled by molting hormone. Now, in this flow chart, you can see the levels of molting hormone. You see the level of molting hormone at the larval stage, adult stage, imago stage. In all these stages, it is produced in equal levels. But you can see the juvenile hormone, once the process of prothoracic hormone, the molting hormone, is properly taking place in the larval stage. It is present in the highest level from the first molt, second molt, third molt, and all the levels of the juvenile hormone reducing. And while it is reaching the imago stage or the final stage to form an adult, the JH hormone is becoming least. Okay, so now there is formation of the proper adult. Okay, so this is the different levels at which the juvenile hormone acts. Let us move on. Now, what happens exactly? Now, we have seen the corporalata, brain, and the prothoracic gland are involved in production of the hormones. Now, the ecdysone is produced by the prothoracic gland. Now, as the levels of ecdysone increasing, we know very well there is stimulation of the imaginal buds to develop into different structures inside the larva. And as a result, the flow of JH keeps on reducing. As there are adult structures that are progressively forming, the activity of the JH keeps on reducing. Reduction in the activity of the JH causes the death of larval cells. And this disintegration or death of the larval cells will be, the dead cells will be used as nutrients for the growing imaginal buds. Okay. Now, if the amount of JH is lower in blood, the mounting from larva to pupa takes place. From pupa to adult JH will be absent. This we have seen in the previous flow chart where we have seen the concentration is higher, it is reducing, and it is least when there is development from pupa to adult. Now, apart from these hormones, there are also what are known as insect growth regulators, which are artificially synthesized. They also can control the process of metamorphosis. For example, the methaprine, hydroprine. These are the analogs of juvenile hormone. They also can be used to control the insect growth. And there are also what are known as the chitin inhibitors. These ones they are used, they can prevent the formation of the chitin. This is used in integrated pest management to prevent the insect pest in the agricultural fields. Now, after coming to know about the hormonal control in insect metamorphosis, let us see how it happens in case of the vertebrates. We are taking the example of amphibians here. Now, metamorphosis in amphibians, it is mainly controlled by what are known as thyroid hormones. Two important hormones are produced by thyroid gland. One is thyroxine, another one is T3 or the triiodothyronine. Okay. Now, in order to understand the effect of thyroid hormone on the metamorphic changes in the frog, if you remove the thyroid gland, the tadpoles, early tadpoles lead to larva never getting metamorphosed into adult. Okay, so absence of thyroid gland never resulted in formation of the adult. Instead, they remained as larvae. They increased in their size and became what are known as giant tadpoles. So this shows that for metamorphosis, it is very important to have proper levels of thyroid hormones. This is a cycle of metamorphosis. You can see the tadpole present here. The tadpole undergoes series of changes to become an adult. Okay, so these changes, development of the adult structures and disappearance of the larval structures occurs under the influence of the thyroid hormone. Now let us see what role the thyroid hormone plays. Now, the role of thyroid hormone, it is different in different regions. Okay, so in the head region it is different, in the trunk region it is different, and in case of the tadpole larva, there is a tail. In the tail region, it is different. Now, for example, that is what I have highlighted here. Same stimulus can cause tissues to degenerate, and at the same time in some other part it can stimulate development and differentiation. At some particular region it can bring about the disintegration of cells, and some other region the same stimulus can bring about development and differentiation of the cells. ಒಂದು ಕಡೆ ಸೆಲ್ ಪ್ರೊಪಗೇಷನ್ ಅನ್ನ ಸ್ಟಿಮ್ಯುಲೇಟ್ ಮಾಡುತ್ತೆ, ಇನ್ನೊಂದು ರೀಜನ್ ಅಲ್ಲಿ ಸೆಲ್ ಡಿಸಿಂಟಿಗ್ರೇಷನ್ ಅನ್ನ ಸ್ಟಿಮ್ಯುಲೇಟ್ ಮಾಡುತ್ತೆ. ಒಂದೇ ಹಾರ್ಮೋನ್ ನ ಸ್ಟಿಮ್ಯುಲಸ್ ಬೇರೆ ಬೇರೆ ಜಾಗದಲ್ಲಿ ಬೇರೆ ಬೇರೆ ತರ ಎಫೆಕ್ಟ್ ಅನ್ನ ಮಾಡ್ತಾ ಇದೆ. ದಟ್ ಇಸ್ ವೈ ವಿ ಆರ್ ಹ್ಯಾವಿಂಗ್ ದ ಟೈಟಲ್ ಆಸ್ ರೀಜನಲ್ ಸ್ಪೆಸಿಫಿಕ್ ರೆಸ್ಪಾನ್ಸ್ ಆಫ್ ದ ಹಾರ್ಮೋನ್ಸ್. ಅರ್ಥ ಆಯ್ತಾ? ಈಗ ಒಂದು ಎಕ್ಸಾಂಪಲ್ ತಗೋಬೇಕು ಅಂತಂದ್ರೆ, now increased levels of thyroid hormone in tail regeneration. Tail region alli, if the thyroid hormone level increases, disintegration or degeneration of the tail starts. Okay, but on the other regions like head and trunk region, there is progressive changes, development of the digestive system, brain, and all. Okay, so under the influence of the thyroid hormone. Now, increase in the thyroid hormone levels in the tail region stimulates the production of these hormones like collagenases and metalloproteinases that brings about disintegration of the tail to form an adult. Now, this is what we were saying. For example, the tadpole head and body epidermis differentiates into new glands when exposed to T3. When T3 is present in the head and body epidermal regions, there is formation of the new glands. But the same T3, when it is present in the tail region, it causes death of the tail cells, resulting in disintegration of the tail region. Same hormone bringing about two different responses in two different tissues. Okay. Now, this regional specificity of the thyroid hormone is due to the response of the epidermis in different regions of the tadpole to the different hormones. Now, this regional specificity is possible by the hormones only when the epidermal response is different. Okay, so the receptors that are present on these epidermal cells, they have to respond differently, whether to get whether to develop and disintegrate, or whether to develop and differentiate. Sorry, whether to develop and differentiate, or whether to get disintegrated. Disintegrated, then the cell will die and that structure will disappear. Otherwise, T3 presence in head and body region ಏನಾಗುತ್ತೆ T3 ಜಾಸ್ತಿ ಆದಾಗ automatically there is cell division. Okay, there is cell division and further there is differentiation of the cell into a specific cell which performs a specific function. Okay, so this is what we call it as regional specific response of the hormone. Now, apart from that, different levels of different concentration of the hormone brings about different responses that we shall see. When tadpoles are exposed to low concentration of thyroid hormone, this only brings about shortening of intestine and accelerated hind limb growth. Okay, so low levels. When there is higher level of thyroid hormone, this leads to tail regression. Okay, and there is proper formation of the hind limb. So this shows that thyroid should not be very high. At the same time, it should not be very low. It should be there in an optimum level. In fact, to say it should be gradually increasing from a larva to a adult. Okay, so this gradual increase in the thyroid hormone will bring about progressive changes, at the same time regression of the larval structures, resulting in a sexually matured adult. Okay, now this thyroid gland, which is controlling the metamorphic changes, this in turn is controlled by another important gland present in the brain known as pituitary gland. Now, there is a positive feedback. Once the pituitary gland produces thyroid stimulating hormone TSH, it stimulates the thyroid gland to produce T3 and T4. Okay, so initially increasing levels further stimulates the pituitary gland to produce more and more TSH. And if you look at the level at which the thyroid hormone is acting, it acts as the transcriptional level. Okay, the thyroid receptors are there on the nuclear membrane, and once it enters into the nucleus, genes are present. Specific genes that are required for the process of metamorphosis are activated by the thyroid hormones. Okay, so that is why we call it as it acts as the level of transcription. In some nuclei, it activates the genes, and in some nuclei, it represses the genes. Okay, so both are important for bringing about a successful metamorphosis. Okay, initially, thyroid hormone receptors are activated, which stimulate metamorphic events, and metamorphic climax is reached once all the metamorphic changes are done with the adult is almost formed. The metamorphic climax is reached by enhanced production of T3 receptors. So this is what we call it as there is gradual increase in the activity of the thyroid hormone to bring about successful metamorphosis. Apart from the thyroid hormone, one more hormone plays an important role during metamorphosis in amphibians, that is the prolactin. Okay, so the prolactin levels are very high in the larval stages. That means in the pre-metamorphic stage, we see in the tadpoles there is very high levels of prolactin that is present, and T3, T4 are very low. It is somewhat similar to the JH hormone which was present in the insects, which try to maintain the larval characters. Now we see once the metamorphosis starts in the pro-metamorphic stage, the levels of prolactin keeps on reducing. Now TSH, T3, T4 levels increase. Okay, so as there is increase in TSH, T3, and T4, there is pro-metamorphic changes, and when the larval stages or the pre-metamorphic stages, the prolactin levels are low, prolactin levels are high, and T3, T4, and TSH levels are low. So if you understand there is low levels of T3, T4, and TSH, that means the metamorphosis has not yet started. But if you see the prolactin levels are low, T3, T4, and TSH levels are very high, it means that the process of metamorphosis have already started. Now at this climax, that means the metamorphic climax, the prolactin levels suddenly increase and it maintains the steady level. Okay, so this is an important point. So suddenly there is increase in the prolactin levels at the climax stage of the metamorphosis, and it maintains the same level that is required. Now the TSH is high till the climax. Why? Thyroid hormone is required for the greater metamorphic changes, and T4, the tetrahydrothioronine levels become low at the end of the metamorphosis, because the function of the thyroid hormone is already done. Now it has to be present in low and required optimum level to bring about the metabolic changes or metabolic processes that has to occur in the adult, because there are no metamorphic changes occurring. It is the level of T3 and T4 now required only for the metabolic activities of the adult. Now, this is summary where we have discussed regarding the hormonal control of the metamorphic changes in insects and amphibians. Remember, in case of insects, the neurosecretory cells of the brain, corpora allata, and prothoracic glands, they produce PTTH, JH, and ecdysone. PTTH stimulated the prothoracic gland, which produced ecdysone, brought about the molting, and JH, it maintained the larval characters and regulated the qualitative changes that has to occur in the larva to become adult. And in case of amphibians, we know very well the thyroid hormone and the prolactin played an important role. Thyroid hormone levels gradually started increasing. That means TSH, T3, and T4, they gradually started increasing from tadpole to the adult. Prolactin levels were very low in the pre-metamorphic stage, and they gradually started reducing as the metamorphosis was growing on. At the metamorphic climax, the prolactin levels suddenly increase and maintain a particular level, and at the end of the metamorphosis, the thyroid hormone levels drop and maintain a level that is required for the metabolic changes that has to take place in the adult, metabolic processes regularly that occur in the adult. Okay, so that's it, students, regarding the hormonal control of metamorphosis. Thank you all.